second MAC-EPID of the academic year. This one on the second and part 2 of climate change and health, microbial threats and microbial solutions. In case you're interested, 181 people have signed up for the symposium. This room seats 100, and but it's hybrid. So about 80 people said they were going to be remote. If you are remote, we have someone minding the chat if you would like to ask a question. And so there is a question box, is that right? And so put your question there and we'll interrupt so that your question can be asked. For those of you who don't know me, I'm Betsy Foxman, and I'm the director of the Center for Molecular and Clinical Epidemiology of Infectious Diseases, and running the MAC-EPID Symposia series has been one of the great joys of my academic life, actually. This, as I'll show you later, is the 37th symposia. We've been doing 2 a year. We started with 1 a year in 2003, and 2 a year seems to be just about the right amount. So I'm just going to tell you a little bit about MAC-EPID before we go into the things you're all waiting for as our guest speakers, but just so one of the things that MAC-EPID hosts is the integrated training program in microbial systems. This is the second training grant that we've had. The first one was NIH funded. This one is funded by Burroughs Wellcome. And both training grants were basically the same format. The idea is to train future scientists who will be able to design and implement novel prevention and diagnostic treatments, strategies that stem from this deep understanding of the impact of microbial communities on health and the environment. And items has been open to all doctoral students at the university, and we have had students from many areas in the university. And the training, it's a independent study kind of training program that has to include certain components. That is training in mathematical modeling, simulation modeling, laboratory methods, and population sciences. And to enhance that, they have to identify 2 mentors covering for sure at least one laboratory sciences and one of the other 2. And they're strongly encouraged to include in their dissertations those different components. And we're very proud of our students. These are some graduates from this last year. You can see here that we have a very diverse set of topics that were covered. James is like characterizing and using a droplet-enabled co-cultivation of elucidate interactions for microbiomes. And Nicholas did systems theory in soil, agroecology, and Frida salivary omics, measurements and biomarkers for epidemiologic analysis. And it's really wonderful to have students come from across campus and find out that just though they come from different academic silos, how talking to people from different areas is both stimulating, and there's much more commonality in doing science in microbial systems than you might think, even if you come from very different departments. The other one of the programs that I direct and also founded is this CHIP program, Certificate in Healthcare Infection Prevention and Control. We are now in our 13th year. We've trained over 70 students to be infection preventionists. It's open to master's students at the School of Public Health. They take courses that prepare them to take the certification exam for being a healthcare infection preventionist. And then there's an internship program which gives them the leg up that you need to get an infection prevention job, because basically if you don't have experience, you won't get hired, and you can't get experience unless you're hired. So it's just really right. Anyway, so wait. This is the 37th symposium. We were counting back and forth, but anyway, forget that. I already mentioned that. But if you want to, it would have been the 38th, but we had to cancel one because of COVID that was all set. If you're wondering how we fit this topics. We have a very serious meeting like you see here. And we ask people to bring forward their ideas. I usually solicit people ahead of time and ask if they had ideas, and ask them to come to the meeting and pitch it, and we'll see what will rise to the top. And then whatever rises to the top becomes my problem. And then I use my network and ask people to suggest speakers. We like to have speakers who are excellent speakers and provocative and interested and want to engage with students and in the pre-workshop activities, pre-meeting activities, and also by reviewing the literature. And I try to find a mix of speakers that represent different aspects of the same problem. I don't know, have the three of you met before? No, and this is pretty typical, that try to get people who work on different aspects and they've never met each other before, but hopefully then they will. And that also enhances interdisciplinary interactions. So the goal today is not just to learn something, but to meet people, to foster discussion and networking, and then to provide opportunities for student training. So you'll notice that the schedule is not like a usual schedule for a scientific meeting. After each speaker, there's a break. We ask people to speak 45 to 50 minutes, and we let questions go as long as needed. We have actually run into the next speaker's time on occasion, but usually there's a break. As you all know, in the ballroom, that's always open. And if you're in the ballroom, if you just find yourself in a great conversation you want to keep an eye on, we are streaming it in there as well, if you want to see what's going on. So there's plenty of time to do that. If you haven't been in this building before, there are bathrooms at both ends of the hallway. You might have to go through double doors at the other end, but there are. So turn right or turn left, it doesn't matter, you'll find a bathroom. It's always nice to know. And so I hope that you take this opportunity to meet with our speakers and meet your fellow colleagues and think about things, and maybe make some new friends and get some new ideas. There will be a lunch buffet and refreshments that are also served across the ballroom, across the hall. So, and this is just a lovely meeting space, so we're really great that we can meet there. And it's also centrally located on campus, and I hope that made it easier for you to get here and park or walk. Was it easier to park, Chris? No. Oh, okay. All right. We have a visitor from MSU. Okay. So, and when you see Anna Cronenwet, please give her your hearty thanks, because she makes this a well-oiled machine that I do not have to worry about. Everything from planning all the travel for our speakers, the arranging for the food, the space, and then all this hybrid stuff. So please give her our hearty thanks, and you'll see her running around. And then if you ever wondered how you pay for such a thing, so we use some money from the training grant, also money from the center, and then very grateful to the Department of Epidemiology, who has made one of their lectureship funds available to support one of the speakers each year. And with that, just to let you know, there will be a fall symposium, right? So you can save the date for November 15th. So one thing I failed to mention is that, I disconnected the external display. Shoot. Okay, someone can fix that as we get up the next speaker. So one thing that we do is we ask each speaker to be introduced by a student, and that's part of the professionalization and training that we make. So I'm sorry, since I managed to make everything escape up here. Oh, and please, and thank Morgan, who is being our webmaster here. Somehow I totally screwed everything up. Okay. And so I can't remember who is introducing our first speaker. Alex. Yes, Alex. Okay, so we'll do that.
Hi everyone, I'm Alex. I'm a PhD student in environmental engineering in Raskin Lab. It is my honor to welcome our esteemed guest, Professor Sandra McLellan. She is a distinguished professor in ecosystems health at the School of Freshwater Science at University of Wisconsin-Milwaukee. She had a PhD in environmental health at University of Cincinnati Medical Center and a bachelor in health science at the University of Wisconsin-Milwaukee. Her career has been dedicated to bridging the gap between environmental integrity and public health. The core of her research revolves around the pressing issue of pollution in urban coastal regions and its dire effects on human health and natural ecosystems. Her laboratory's work is pioneering in this approach to understanding how pathogens carried by stormwater runoff and sewage overflows find their way into freshwater systems like the Great Lakes. This research is crucial as these bodies of water are not just ecological treasures, but also vital resources for drinking water for millions. Her research informs policy making and safeguard public health. As we dive into today's discussions, her insights promise to enlighten us on the vital connections between environment and well-being of human health. Please join me in welcoming Professor Sandra McLellan.
Thanks, Alex, for that great introduction. And I really want to thank Dr. Foxman and Dr. Raskin for this invitation. It's been great to be here. Excellent discussions and presentations yesterday, and it's been fun to get to know two new colleagues. As Alex has alluded to, I'm going to talk about the microbes that inhabit our city and the human microbiome, and then the intersection of how that gets into our water resources and how climate is really a big driver of some of these problems. So for today's talk, I want to start with the urban water cycle, kind of the infrastructure, how it's built, the vulnerabilities, aka Sewers 101. And then talk a little bit about the human microbiome and how we've used that to develop new indicators for fecal pollution, such as agricultural runoff and sewage. And how we've been able to track kind of the pulse of sewage that goes into our waterways every time it rains. And then I'd like to kind of wrap up with kind of our more recent research where we're actually looking at the pipe microbes, these microbes that are residents within pipes that were kind of unexpected to find them there, and how we're using comparative genomics and some really new bioinformatic tools that I want to share with you to look at how they are possibly contributing to infrastructure failure. So many of you are familiar with the urban water cycle. It starts out with drawing up water from a source, treating it, sending it through our city, returning it as wastewater, going to the wastewater treatment plant, and then hopefully returning it to the environment pretty clean. Between this cycle, we have kind of a short circuit on our wastewater system called combined sewers, where sometimes when it rains a lot in the city, they can overflow and that water goes directly into waterways. And then we also have stormwater systems where it collects water directly from the environment, and it's a 4-lane highway into the rivers. It's these pipe systems that I'm most interested in. There's been so much attention paid to kind of the microbial ecology of what goes on at wastewater treatment plants, but yet we have 10,000 miles of pipes with this huge biomass, and they're really underexplored as far as their metabolic potential, both positively, if breaking down waste, and negatively, maybe how they're corroding sewer pipes or being released into the environment. As I started moving from maybe clinical microbiology into environmental microbiology, I've learned a lot about sewers and the history of sewers. So for 3,000 years, we've been finding ways to build ditches and send waste away from our settlements. But it really wasn't until the mid-1800s that we started building modern sewer infrastructure. And these are just two great snapshots from London, 1958, and Paris, 1858, and Paris, 1878, where these were the modern marvels of the world. Big, huge sewer pipes that were taking waste away to the rivers. And they were such a marvel that there were tours of them in Paris. If you go to Paris, you can still go on a sewer tour to this day. It was not, well, it was a little surprising to learn that these pipes went right to rivers, and it wasn't until like the late 1800s, early 1900s that we thought to connect them to wastewater treatment plants. So the first treatment plant in the US was built in 1890. So when we look around at these older cities around the United States, they're combined sewer systems because, again, they were originally built just to pipe everything away from the city. Rainwater, the stormwater, along with the waste from people and buildings. And then these pipes were connected to wastewater treatment plants. When it rains a lot, these pipes fill up and it can damage the microbes that are performing treatment in these treatment plants. So they're designed to actually overflow and release that rainwater along with sewage directly into rivers and lakes. And we find these systems in 772 communities around the US. They're really concentrated in the Northeast, Midwest, and Pacific Northwest, which are, of course, the older cities around the United States. So in all, there's about 850 billion gallons of combined sewage that's released each year. And being on the Great Lakes, if you look at this map, many of these systems are in the Great Lakes drainage system, and that's going directly to a drinking water source where the Great Lakes are a drinking water source for more than 40 million people. Past about 1920, we started building separated sewer systems. So when you go into any city and you get to kind of that outer ring where you start to see those 1940 houses, 1950 houses, you start to see storm sewers around the area that discharge directly to rivers. So this is good in that we are less susceptible to combined sewer overflows, but it's bad because everything that washes off the street is going directly to rivers, untreated. So again, as a microbiologist, I've kind of taken a crash course in environmental engineering. I joke with my environmental engineering colleagues that I slept in a Holiday Inn last night. So I'm not an environmental engineer, but I've learned a lot from them. And one of the most important kind of mechanisms that explain some of the contamination we see is infiltration and exfiltration. And this cartoon on the bottom here kind of describes it, where if you have a sewer pipe, it's 100 years old, it's leaking, things can leak out. But as the water table, so that's exfiltration, as the water table rises, maybe through rainfall, or if you're in an area with a high water table, you have infiltration. It comes into the sewer pipe. So most environmental engineers at wastewater agencies are worried about this last panel, infiltration. They have to treat all that rainwater. How are they going to, handle that water when it rains? And they worry about their treatment plant keeping up. But we're actually worried about it on the other side, the exfiltration, because what happens is the leaking sewage gets into the soil around it. But then when it rains, it can migrate and go into stormwater pipes that also are incredibly leaky. So we see the sewage signal in stormwater pipes, and we know that there's kind of this soft connection between pipes. So we think a pipe, it shouldn't be leaking. Well, when we think about our cities, some of these pipes are like 100 years old. I'm working with a project in San Diego, and they're trying to kind of quantify how much exfiltration, and they have groups of pipes, and they have the PVC, and then they have clay, and they literally have some paper mache or brick sewers that they're looking at. The EPA estimates that this is a $1 trillion problem. It's common to every city. Every city has leaking infrastructure. And I always think, the city will fill a pothole in 24 hours if you call the hotline, but we really don't understand what's going on under the street until we start seeing these signals coming out of stormwater outfalls. So how is climate going to affect all of these problems? I told you about two vulnerabilities: CSOs overwhelming the combined sewer systems, and also stormwater runoff. When we look at how this is all driven by rainfall, and when we look at how climate is going to change, we can see that there's going to be more extreme precipitation events, particularly in the Northeast, Midwest, and Pacific Northwest, which unfortunately kind of parallels where these combined sewer systems are. So not only do we have vulnerable infrastructure, it's the most vulnerable for the extreme events. So for 100 years, we've tracked fecal pollution by following either fecal coliforms or E. coli. That's how we figure out sewage, agricultural waste, something's in our waterways. So it's a really good indicator if you want to know something's there. It's easy to culture. It's kind of low tech. Almost any lab can do it. But the problem is it does not tell you where the source of pollution is coming from. So you can go monitor a river and find E. coli for 40 years. But what are you going to go fix? Are you going to go upstream and look at the agricultural runoff? Is it just a stormwater problem? Do you have a lot of dog waste and wildlife going in your stormwater system, or do you have leaking sewers? And if you look at E. coli, you can't tell the difference between it. But it's really important to know that difference, not only for what to fix, but what's the risk. I am not so worried about what comes from dog waste as I am from what comes from sewage, which is really where the reservoir of a lot of human pathogens are. So this is just kind of a quick table of some of the pathogens we worry about in fecal pollution. The orange panel is mainly animal and agricultural runoff. So we have things like the E. coli 0157s, Campylobacter, Salmonella, some of the protozoa. And then when we think about what is the risk for sewage, it's mainly the viruses, and norovirus is the one that, you know, is really hardy. And I see Kate nodding like, yes, norovirus is a problem. The norovirus is probably the most pretty hardy in the environment, but the most infectious. It doesn't take much. So when we look at swimming risk, mostly you're worried about norovirus, but some of the other enteroviruses also can be really severe. And among the enteroviruses, you know, I wanted to call out hepatitis A. So my colleague Justin Seymour and I hopefully have, fingers crossed if the reviewers like it, a review that covers just how climate is going to modulate some of these infectious diseases. And we hope that review will be out this spring if we can get past reviewer 1. So when we think about tracking E. coli, this is just kind of a summary of what we see in our area. And this is a snapshot of the downtown Milwaukee area. For those of you who have been there, you'll kind of recognize we have rivers that come down into our downtown area. All 3 rivers come together through a small 40-meter channel out into the harbor and then into nearshore Lake Michigan. So we have the Milwaukee River, Menominee, and KK. And the EPA limit for recreational waters is 230 E. coli per 100 mils. Above that, they say it's not safe to swim. You have a risk for illness. We see levels of almost 20,000 E. coli per 100 mils when it rains, and this is no known sewage overflow. So very high E. coli. And the question is, where is that coming from? You know, is that agricultural runoff? Is it just stormwater, or do we have leaking sewage? I want to point out we see very high levels also at beaches. And we find these small localized levels are sometimes a stormwater outfall problem, but often just birds roosting on the beach. So for years, people are asking us, why are the beaches closed? Is it, you know, is it agricultural runoff? Is the sewage treatment plant not doing their job? And when we go 10 meters offshore, we don't see any E. coli. We're like, yep, it's the bird waste on the beach. So E. coli is a problem for understanding where sources are coming from. And that's where we got really interested in using the human microbiome, because the organisms in the human microbiome are very specific to humans. And I'll show you some of that data. So we've used that to really track kind of pollution signals in the environment, all the way from the stormwater outfalls through the rivers and into the estuary and the lake. So when we think about the human microbiome, what we're culturing out is a facultative anaerobe. So we can easily grow it, but that's not what our gut usually carries. It's really only about, you know, E. coli is really about less than 1% of our microbiome. 95% of it are fecal anaerobes. So those are not easily cultured in the laboratory, but they are easily detected using molecular methods. So for many years, we have sequenced sewage and other fecal pollution sources. And this is just a figure from one of our more recent papers showing 71 different cities. And this map kind of shows where some of these sites are. We've sampled a lot of cities in the south and the north. We did that over 3 seasons, January, April, and August. We sequenced all of those samples, and they pretty much all gave the same overall profile of who's there. And what we were surprised with was that only about 15% of them are actually from people, are actually fecal microbes. The rest of them were environmental microbes that were very specific to the pipe environment. And this was, I think it was about 2010. At first we thought, what is going on with this data? And then it starts to make sense. Of course, we have a giant chemostat under our cities, a 10,000 mile chemostat, giving it, it's dark, there's water, there's nutrients. Of course, we're growing microbes there. So I first want to talk about the human microbiome. So just the slice of 15% of the organisms. And the main ones we kind of see in the gut are, you know, the Lactospiraceae, the Clostridium, and the Bacteroides. So we did a lot of sequencing, and we tried to find, you know, sequences that were specific to different sources. So this is just a network analysis of some of the major fecal microbiome taxa. And we did a network analysis that connected each sample here, which is labeled, and their sequences with the sources that are shared. And then in the network analysis, we colored it by pink if they were shared by many sources, blue if they were only shared by 2 sources, and then green if they were unique to a source. So we're really interested in all these green sequences and taking them and then developing new indicators. So we have several papers where we've done that. I just want to point out 2 of them. One of them targeted a Lactospiraceae, and we developed a qPCR assay. We call it LACNO3, which is cleverly the 3rd most abundant Lactospiraceae in sewage. So why didn't we do LACNO1? It was one that actually seemed to grow and propagate in the pipe, so it wasn't very indicative of humans. LACNO2 was shared with cows, but LACNO3 we didn't find in any other animals. So it's really abundant, but it's also specific. And then another approach we took was to use random forest to kind of take the whole community of Lactospiraceae or the whole community of the Bacteroides and use random forest to develop a classifier. So there's a paper, actually 2 papers, one using random forest to develop the classifier, and then forensic, where we put the classifier online. And it essentially just takes the profile of what you expect from a source, and it asks if your unknown source has the same proportions of those sequences that you're expecting in the profile. And through that, we've been able to really characterize some of these contaminated waters, both quantitatively with the qPCR, but then also more holistically looking at, is there animal sources? Is there human sources? So I want to show you some of that data. And again, this is the study area that we mostly do our work in. We're in the Great Lakes drainage basin. So all of this dark brown area here is what directly goes to that drinking water source. I'm located in Milwaukee, Wisconsin, and this is the slice of the watershed that's right along this margin. And again, the downtown area, I showed you that snapshot where the 3 rivers come in. And I want to point out the Milwaukee River goes way up into the watershed. It's almost 850 square miles of drainage and a lot of agricultural runoff. The Menominee River is a little bit more suburban. We get a lot of stormwater runoff. And then the KK River is a really urbanized watershed. And if you want to do track pollution signals that go into coastal waters, I couldn't be more perfectly located for a field site, because conveniently Milwaukee sends those 3 rivers together through that channel and into Lake Michigan. So we've sampled the rivers, the harbor, and then we go out by boat into Lake Michigan. When we go out into the lake, the pollution signal gets really dilute. So usually we have to filter about 10, maybe 20 liters of water. But when we're in the rivers, we really only have to filter about 100 to 200 mils to really see these signals. And then I talked about rain really drives us, and it's the extreme storm events that we're worried about. This is just a satellite image, and you can actually see, this is down in Racine. This is the Root Pike coming in. This is Oak Creek. And then you can barely see Milwaukee because it kind of mixes with the harbor before it comes in. But these plumes of river water are actually visible from space. So we took our indicators that we developed, and we went to go look at kind of the footprint of pollution in Lake Michigan. And when we go out and do these kind of mass field surveys, under kind of just rain events, we can track these human specific indicators. These are our PCR markers, both Bacteroides and the Lactobacillus, almost 2 miles out. When there's a combined sewer overflow and heavy rain, we can track them up to 5 miles out and at pretty high levels. So this is about 10 to the 5th per liter of copies that we can detect in these waters. You can't see it because it's pretty dispersed at this point, but it's amazing that we can still detect these indicators. So we've tried to kind of quantify this pulse that comes from the city out into the lake. And we've deployed what we call ISCO samplers, and I'll show you a picture of that in a second. Automated samplers at each of the 3 rivers, the Milwaukee, the ag one, the suburban one, the highly urbanized one, and then one right at this channel before going into Lake Michigan. And we can follow these markers over the storm event. So this is just a picture of what the sampler looks like and a nice little housing on the channel. It fills bottles once every hour for 24 hours. The top panel here is the hydrograph of the water coming in. So we partner with USGS and they can actually measure how much flow has gone by the stream meter. So the very top one is that estuary site where the 3 rivers meet. And then we have the Milwaukee River, which is the highest flow, the Menominee River, which is the second most flow, and then the very small KK River. On the next panel, it shows the first couple days after a storm event. You can see that within a day, we see the human Lactobacillus marker, our lacno3, and human Bacteroides spike at very high levels in the first 2 days. That's that pulse of kind of leaking sewage that's coming in. And then as the ag runoff starts to come in, we can see that pulse of ag runoff show up about 2 to 3 days later. And then this is just a second storm event down here where it just shows it's a pretty reproducible. When it rains, we see this pattern over and over. So for 40 years, they've seen fecal coliforms and E. coli in the channel. Where is it coming from? The answer is multiple places. And we want to kind of quantify then how much is really coming from each source. So working with USGS, we have the flow, we have a concentration in a bottle, so we can do load estimates. And Haley Olds, one of my former master's students, just did this amazing project. I think it was almost 3,000 water samples. And she was able to show the load when there's base flow, the total Lactobacillus copy number. This is just kind of the PCR results. They're about the total number 10 to the 11th, 10 to the 12th. When there's heavy rain, it goes up 1,000 to 10,000 fold. On some heavy rain events, and you can see it's maybe about 3/4 of an inch higher than this other heavy rain, we get up to 10 to the 16th. So that's 100,000 times over what we see on base flow. That's the total amount going into the lake. We've seen this pattern repeat over and over, and it almost seems like there's a tipping point in our sewer systems that maybe there's unrecognized sanitary sewer overflows or something is going on that it's not recognized. Because again, this is not a reported combined sewer overflow. When we do have a full combined sewer overflow, we see kind of similar levels, maybe about 6 to 7 times higher, but in the order of kind of 10 to the 16th total copy numbers going into the lake. And this is just an aerial photo of what it looks like when it's going into the lake. So we can explain combined sewer overflows. We know where all those outfalls are, but what's going on when there's heavy rain? Why is there so much sewage going into Lake Michigan? It gets back to that infiltration and exfiltration dynamic that the environmental engineers know so much about. They worry about infiltration into their sewer pipes. And again, we're kind of looking at that exfiltration and then migration into the stormwater system. So we've worked with our Milwaukee Riverkeeper for more than 10 years now and have sampled hundreds, 300 outfalls up and down the river. And next time you're walking along a river, I'm sure you'll start noticing all these stormwater outfalls now that I've said sewage is probably coming out of them. But this is kind of in the Menominee River watershed, and the red dots are ones that had persistent sewage signals. And then the yellow dots are ones that had more intermediate sewage signals, because again, this is not a sure connection. This is, you know, a rain dynamic. So it takes a certain amount of rain to really push things out. So in a nutshell, to kind of, you know, give you the overview of the 10 years of data, we've done 315 outfalls, 2,000 samples, and we found that 30% of them have a consistent level of untreated sewage. And this is very common for many cities. It's not just our cities. About 10% of them have really high levels, almost looking like a combined sewer overflow. And then interestingly, if you only look at the E. coli data, about 50% of outfalls that would be flagged by, hey, I have high E. coli, have no sewage signal. So if the municipalities are using E. coli to determine, you know, the stormwater impact, they're missing the boat about 50% of the time. Half the time, they're probably just looking at dog waste, bird waste, and the other half of the time, they're maybe seeing a problem. So we're working with like the city of Milwaukee and surrounding communities to try and use these new indicators as the screening process. So then they know where they have to go up the pipe to look for problems. And just a little bit more doom and gloom before I leave this topic about stormwater. I've looked at the American Society of Civil Engineers infrastructure report card for at least 15 years now, and consistently I see the grade D and D plus. I see some of the engineering students nodding into in the so really, you know, we do have a problem keeping pace with our infrastructure, and climate change is going to pressure it more. So we just need to up the pace. And these sewer pipes, some of them can be on a replacement cycle, meaning how much money do I have before I can go fix these pipes on a 150-year cycle. So we need better ways to find them and fix them before they hit the maintenance. Okay, so I want to move into talking about these this other group of organisms that we were kind of initially surprised to find, but then not so surprised. Kind of the pipe residents, and this is what I call them pipe microbiome. And these are the guys that we think are maybe doing some pre-treatment of waste, but also maybe degrading our infrastructure. So we've gone back to our data set of 71 cities that were sampled in January, April, and August. And instead of just looking at the human microbiome, we've pulled out all the sequences that look like they're specific to pipes. And we've kind of dug into what are they doing and what does that look like? So we have a paper, The Guts of the Urban Ecosystem, Microbial Ecology of Sewer Infrastructure. I did not come up with that title. My brilliant colleague, Marut Aaron and Ryan Newton did, but it really, these organisms really reflect what kind of goes on in the human gut. It's almost like we see parallels in our city. The three most high abundant ones consistently in every sample we've ever looked at is Archobacter, Acinetobacter, and Aeromonas. So those are the genus, but when we get to the sequence level, we start to see some really interesting patterns. So I want to walk through this figure that was in the paper. Again, we sampled from 71 different cities, and we can color code these cities by warm cities, kind of using the mean average temperature for the month that it was sampled, or cold cities. And we've kind of organized the samples by the cold cities up here and the warm cities. So each kind of individual column here is one sequence. And we took the top 200 sequences. If I put all 800 up here, we wouldn't have been able to look at them. So what we found when we looked at, when we kind of did a analysis of how they clustered together, we found that there's a group of sequences that have kind of the same occurrence pattern in cold cities. And these sequences are not very common in warm cities. And then when we look at the warm cities, which are kind of down here, we see a whole group of sequences that are common, but they're not very common in the cold cities. So we call them kind of this high temperature profile sequences and these low temperature profile sequences. And what's interesting, you can see a lot of them are proteobacteria. Some of them, there's one or two bacteroides. They're not human bacteroides. They're ones that live in the environment. But when we look at like Archobacter, we see warm Archobacter and cold Archobacter. When we look at Aeromonas, we see warm Aeromonas and cold Aeromonas. What was really interesting in this analysis is when we look at this group of organisms that had no temperature profile whatsoever, they were the human microbiome ones. They were the firmicutes and the bacteroides. So that really told us we were seeing a real signal, because we don't expect the human microbiome to be driven by temperature. We expect the pipe residents to be driven by temperature. So we have a whole group of organisms that tell us there's kind of this ecological signal. Okay, so one of the organisms we're most interested in is the Archobacter. So we've done a lot of work isolating out Archobacter from different places where I have colleagues. I work with one colleague in Spain, Maria Figueres, and to speak to the importance of going to conferences. I went to a conference for a completely different purpose. I was talking about our human microbiome stuff, and this was back in, I want to say it was maybe 2011. But I had one little slice of the pie that was Archobacter. And after my talk, she came up to me and she said, Archobacter, nobody talks about Archobacter. We think it's really important. And now I've collaborated with her for more than 15 years. And she's really gave me a lot of insight and shared her expertise on this. So we've been culturing out Archobacter from Spain, from Wisconsin, from Hawaii, from Australia. And we want to know, even though we see these temperature patterns, but are there geographic patterns? I mean, think about Europe, Wisconsin, Hawaii, and Australia, very geographically separated, right, by oceans. And this is a picture of my colleague, Justin Seymour, who is my collaborator in Australia. So one of our main questions was, are the organisms the same in Hawaii, Australia, Wisconsin? And just to show you, I like to visit my collaborators because Wisconsin sometimes looks like that, or how it's snowing today. Not only have we isolated these organisms, we also have sewage samples where we've done the 16S community data from 17 different cities around the world. So we can kind of ground truth what we see in the genomics with some of the other community data. This is just a table of the numbers of isolates I have. So we've sequenced about 117 of them, and now we're starting to really analyze them. And I want to put a plug in for the platform I use for the comparative genomics, but it's also great for metagenomics, Anvio. And it's been an open source platform that's been developed for, I want to say, almost 15 years now by my colleague, Marut Marin, and he's at the University of Oldenburg now. But this platform is not a pipeline to analyze data. It's like your kitchen, right? It gives you all the tools. So if you come in and want to make dessert, you can make dessert. If you come in and want to make a smoothie, you can make a smoothie. If you want to cook a steak, you can cook a steak. So it puts the data in this amazing format where you can come in and ask all of these different questions. So the workflow in Anvio for pangenomics is you really take your genome sequences and you make what's called a contig database. And it's just, I'm not sure what goes on behind the scenes, but it puts it in a format that it's not just sequence data, it's captured as a database. And then you take these individual sequences and you put them together in just a group called genome storage. It's like putting all your clothes in a single closet. And then from that, you can essentially use one line of code to do pangenomics. And I'll show you some figures of that. Or you can use one line of code to pull out 16S sequences, or one line of code to do functional, enrichment. So we use Anvio quite a bit. And I wanted to put in a plug for this because even as a PI, I can use Anvio. It's really a great, like I said, open source platform, but a great community where they're on Discord. So if you have a problem, you can just post something and somebody will answer like literally within 5 minutes. So we use Anvio to really look at these geographic signals and can we find geographic signals? So this is just kind of an overview of what Anvio output looks like. And we have each genome is kind of one of these circles, and I've colored them from where they are from. So the pink ones are from Australia, the green ones are from Hawaii, the blue ones are from Wisconsin, the green ones I pulled out of GenBank. There's some orange ones that I pulled from pipes. And then you take these genomes and you align all the gene calls. So this dark area is where all the core genes are. Every genome shares this. And this kind of scattering over here are where the accessory genome is, genes that are shared by some organisms, but not all organisms. And then there's some kind of low frequency accessory genomes. So I can take this kind of gene frequency and I can ask Ambio, make me a tree based on gene frequency. So Ambio will do that. And then I can start to explore it. So when I look at this phylogenetic tree, and I know it's not blown up here too well, you can see that the Archobacter, and the black ones are just pulled from GenBank too. So from non-sewage sources. And that bar right down here is kind of sewage versus non-sewage sources. So those lighter colors mean that they came from sewage. But we start to see clusters. And it was really interesting that Hawaii and Australia genomes on the basis of, shared gene frequency clustered really tightly together. And they're separated by a 10-hour flight. The Australia ones, a few of them also clustered with the Milwaukee and GenBank ones. And then the pipe ones, the ones that we scraped from the biofilm, looked really different. And these are all within the genus Cryophilus. So this is a single genus. So now we started to see, yes, there is a geographic signal. That's really interesting. But what are they doing? What's some of the functions? And one of the functions we found common to all Archobacter cryophilus was the sulfur cycle and sulfur oxidation. And this is just a nice schematic from a paper from 2020 from Wu et al. That just shows what happens in sewer pipes in the sulfur cycle and why it's bad. We put a lot of waste in. There's a lot of anaerobes in the pipes. So they start to, they lack oxygen, they use up oxygen really quickly. They start to use sulfur as an electron acceptor. They make H2S. The H2S can dissipate into the headspace, into the biofilms, where there's sulfur oxidizing bacteria. And then that makes sulfuric acid and it starts to corrode the concrete pipes. What's interesting about Archobacter is we find it, those were the pipe ones, the orange ones, we find it on the biofilm, but we also find it planktonic and in the sediments. So how is that cycling back through not just the headspace, but these other parts of the pipe system? And that's something we're currently working on. So this is just a snapshot too of the sulfur cycle. So what's really cool about Ambio is, how did I figure this out? How did I figure out where these genes are? Again, I'm the PI. I'm not the expert in the lab doing the bioinformatics. But because I have all my genomes in this genome storage and I can query them, I can go into Ambio and pick a pathway. So I go into KEGG and I pulled up 2 of the sulfur pathways that I was interested in, and I know their numbers. And I literally go in Ambio and I say, find me these genes. And it will find these genes for me. And this is just a little blow up of how it will annotate these gene bins. And you can really zoom in and actually look. These are all core genes, but when they become accessory genes, you can zoom in and see it's in this organism and not in this other genome. So we're in the process of trying to figure out the role of these sulfur oxidizing genes in the planktonic part of the pipe or the sediments. But this is where it all kind of came together actually just in January about this geographic signal. So in Ambio, again, with 1 line of code, which is shown here, I was able to pull out all the SOX genes, and I pulled out the regulators. I pulled out all 7. I concatenated them, and I said, make me a tree of just the SOX genes. Let's see how the genomes are related, not on all the genes they own, but just the genes involved in sulfur. And it was really cool to recover this tree of, there's the Milwaukee isolates. They have kind of diverged SOX genes. There's the Hawaii isolates and there's the Australia isolates. And that's just on the functional gene. So it was super cool. And we're excited to then dive into pretty much all these other organisms, not just the Cryophilus species, and many other kind of metabolic genes that might be participating in this metabolism. So again, my last plug for Ambio, you can do things like look for enrichment. So again, with 1 line of code, I can say, here's genomes from sewers, here's genomes not from sewers. Tell me functionally what's enriched in group A versus group B. I can divide it by Australia. I can divide it by, pipe bacteria, biofilm versus not biofilm, and figure out what's different in these. So just to kind of wrap up the pipe microbiome and how it relates to climate, because this is all really cool. This is how pipes work, but what's going to happen with climate change? So I told you about the extreme rainfall. That's going to happen in spring, probably for us. But I don't know if any of you have noticed, but sometimes it doesn't rain in summer now. And that's a big problem for sewer systems because that's how they get their aeration. And that's how they get out of that anaerobic state. We actually have a new project with our Milwaukee Metropolitan Sewage District to look at these microbial communities in summer, because last summer, their sewage going into the plant was so septic, so anaerobic, that the plant couldn't handle it and they were going out of compliance. So they're seeing this very practical outcome of lack of rain, lack of aeration. And again, it's linked to the sulfur cycle. So if I just pulled a couple snapshots from the NOAA site, climate.gov, which is just an amazing source for, looking at how things will change. And these are just 4 random summer days, just to show you that drought can happen anywhere in the country. So it's not just the South or the West that's going to be having these problems in their infrastructure. It's pretty much everywhere, except for the poor Northeast. They're just going to keep having combined sewer overflow problems. But the problems are kind of twofold. They're immediate, the summer of the drought, where you're getting septic sewage going into the plant, and do they have to do some engineering control? Do they have to flush their hydrants? I mean, do they have to, ask people to water their lawns into their sewers? But also they can be contributing to the sewer corrosion because you have that sulfur cycle now going much more efficiently because it's not anaerobic. So I want to wrap up with coming back to kind of the imprint on coastal waters, because I showed you this picture of all the human microbiome and how we can see it way 5 miles out into the lake. When we look at these pipe bacteria, which again are the major portion of the microbial community, and these Archobacter can be up to 15% of the community, we can see the Archobacter signal in the community all the way out to 5 miles out, but it's not just doing gene copies. We can see it's 1% of the microbial community. So we are taking these non-indigenous kind of microbial weeds, right? They're like a lot of heterotrophic bacteria, and we're putting them into the lake in these pulses. So Ryan Newton, my colleague, is really interested in how these non-indigenous bacteria are going to affect the lake ecosystem. So just a few conclusions. As I wrap up, leaking sewers and sewage overflows are really what sends pathogens into water, and the increasing storms are going to be a problem, especially in the areas I showed you. And this is where I think the new indicators are going to be really important, because we don't have enough money to fix all of these problems. We really have to do a better job protecting public health. So not just closing the beach because there's E. coli, but really closing the beach when there's sewage there. And by the way, E. coli might not even be elevated in those cases, and also use them as find and fix. We can't fix every pipe, but let's target the ones that are the biggest load into our waters. And then also we have 10,000 miles of pipe below a city like Milwaukee, even more in New York and the major cities. And it's a huge metabolic potential, both good and bad. And I think we're just starting to unravel that and appreciate what we call like biological infrastructure, which might be just as important as kind of the hard infrastructure that we're putting all these investments in. And again, we just, climate change is going to make things even more challenging in the future. So I'd like to really acknowledge my lab. A whole lot of people have worked on this, but especially 2 former postdocs, Adelaide Rogeau, who did the guts of the urban ecosystem, and Shushen Fang, who did all the work with the Lachnospiraceae and developing these indicators. And we've been really fortunate to be funded by a diverse array of funding sources from NIH all the way to our Wisconsin DNR and our sewage district. And then especially my collaborators in hydrology, Steve Corsi, and my collaborators with Arcobacter, and some of our early work with Mitch Sogan, who really got the ball going with some of his sequencing. So I'll end with that, and I'm happy to take any questions.
If you have a question, if you can use the microphone, so that way the online.
Hi, thanks so much for your talk. My question is, do you have a sense, like in specifically the Milwaukee watershed, like how much, are there any like septic systems? And also, do any of these indicators that you're looking at do anything to distinguish like urban wastewater from like maybe what might be coming from malfunctioning septic systems?
Oh, that's such a great question. We don't have a lot of septic systems, but Michigan and my colleague Joan Rose has looked a lot at that. And we have done it just a little bit, but in Hawaii. So for those of you that visit any tropical island, there's not a lot of good sewage systems. And most of them are cesspits and cesspools, and they actually overflow. So we do see the same kind of similar organisms like the Arcobacter and all that, but the human microbiome ones are a little harder to track because they're from individual people. So not everybody will carry the same amount of lactobacillus or Bacteroides. But in general, both types of indicators, I would say it's more patchy data, but we still see the same signals. Yeah.
I'm glad that you love the neglected epsilon proteobacteria. So as an infectious disease physician, I always considered them the kind of poor stepchildren of the helicobacters and the campylobacters, but they do cause disease. In your analysis of comparative genomics, did you ever seed in some of the ones that have been isolated from HIV patients or other patients with gastroenteritis and see how they fit with the ones that you find in the pipes?
Oh, that's a really great question. I'd love to talk with you more about that. I did not show any of the Arcobacter butzleri because they do not have the sulfur genes.
Well, also causes.
Right. Yeah. So yeah, we haven't, they are in kind of our genome comparisons. We haven't looked specifically at the pathogenicity kind of genes versus the sewer ones. That would be a great thing to look at. We've mostly looked at the butzleri, and we see they're really distinct. They're kind of low abundance in our system, and we don't see the pathogenic ones very much. I'll have to look specifically at the Caraphilus pathogens. They mostly come from like the isolates that are in GenBank are mostly from aborted fetuses from cattle or whatever, and they're not in the same cluster, but it would be really easy to put those in one group and the sewer ones and do that metabolic enrichment or functional enrichment to see what is different about them.
I think part of the problem is that we used to see them earlier in the HIV epidemic before we were like sequencing everything. The idea of getting a genome was like we would joke about it.
Right.
Oh yeah, let's just sequence the genome. Ha ha ha. But so they're not going to be in the databases, but I think they might be in some people's freezers.
Oh, these would
be like the old boxes that everyone's, can we throw these away, Vince?
I know. Yeah. And then you're like, where is that box? Yeah. That's a really great point. And we did approach NIH once, talking about butzleri and some of the foodborne implications, but I haven't thought about specifically looking at Caraphilus. I might have to catch you on the break on talking about that one.
Thank you so much for that talk. That was pretty great. I think that we had talked a little bit yesterday, and of course I have some questions about biofilm. So I didn't, and the schematic that you have of the sulfur cycling within the pipes, it looks like the biofilm almost is like protecting the pipe where it isn't being corroded. I didn't know if that is something that people are looking at whatsoever, or if that is actually truly a signal.
I think it's the biofilm actually doing the conversion and sending the sulfuric acid in contact with the pipe. So I think the corrosion part is really tied to what's going on in the biofilm. But that's an interesting concept of, you know, can you really promote biofilm that doesn't have that function?
Yeah. Yeah, because there's so many layers to a biofilm. It seems as if that conversion might be happening at the interface with the water, but the interface with the pipe is actually being
Right.
protected because they're probably, I guess, reducing the sulfur to the point where it's not actually corroding. I guess I don't know if people have actually looked at those biofilms to see.
Yeah, we're just starting to look. I think we've been most interested in what is this huge biomass in the sediment and the planktonic work. So we just kind of tie in the biofilm literature, but it would, I think down the road, it would be great to try and get, you know, some kind of quantitative estimate of where is the sulfur cycling really happening? Is it mostly in the biofilm? By the way, the biofilm guys are distant cousins to Archobacter, the guys who are doing their epsilon proteobacteria, which now is a new name somewhere, like, and next week it will be another new name. But so they are kind of related, but they're distinct. And I think the distinction might be their ability to form biofilms, whereas Archobacter are clearly these modal guys that like to swim around. Yeah.
Yeah, and I didn't know if that schematic was like a true schematic of what it might
Yeah,
look like.
that was from a review paper on kind of sewer corrosion, and they've really kind of pinpointed, like I said, the headspace and also that margin where that water is, of where most of the activity is causing the corrosion.
Yeah, and it almost looked like it was where the water changes level.
Mhm.
And that there is biofilm there. But it dries
Maybe
out.
maybe it's corroding. And I've thought about that because Archobacter is so planktonic and modal, and that's where the oxygen would be available. And I don't know if I mentioned this, to add insult to injury, not only do they do the sulfur oxidation, they're microaerophilic. So nobody else can really work, but they love to work at low oxygen levels. So it kind of starts to make sense in this environment why we see Archobacter.
Really cool. And then I had a question about one of the questions you already kind of addressed, that someone's working on the plume of the microbes that are going out into the lake and the effect on I'm guessing the biome of the lake itself, which is great. That was the big thing that I was wondering about when you're talking about it. And I was wondering if you'll be looking at what you're planning on looking at, I guess, in general. Is it like community composition based off of like a 16S sequencing, or would you be looking at kind of metagenomic data of whether there's gene transfer? Like it's not just the microbes that are being transferred, but also the genes that are.
Yeah, you know, that's a really great question, because Ryan Newton, my colleague, and I talk about our overall approach a lot, and we've really moved towards comparative genomics to really get at the genomes, and then use metagenomics to map what are we missing, or how do we connect this to other environments. But you know, everybody went from 16S to metagenomics, and you know, I'm kind of slow on science, so I just went right to genomics. And I've only put my toe in the water in the metagenomics because it's too complex for me to ever assemble anything. And that's really been insightful because the organisms we're interested in are culturable, and many of the lake ones also are very culturable. So I would say the approach would be, you know, get the genomes and look at what they're doing functionally, and then try and make sense of the metagenomic data. But for the plastics question, it would be really cool to see if plastics near cities get colonized by these kind of urban infrastructure bacteria, and what plastics out in the middle of the lake might get colonized with, which aren't exposed to kind of the same community.
And there's definitely work on that. That yes, there's the ones that are closest to urban areas have these fecal and pathogenic microbes in the biofilm, and the ones that are further away have less. But we don't really know what is it the colonization that's happening in that spot, and it's being lost as it goes out into the lake, because pretty much all plastic is, terrestrial based. So
Right.
the actual, I guess, mechanism of how that we're seeing that pattern is what people are looking to now. So yeah.
You should look for Acinetobacter and Archobacter too.
Oh, I will.
Yeah.
Yes. Thank you.
Yeah, thanks. Yeah.
Very nice talk. So you showed that map with the Northeast and the Pacific Northwest being like the problematic centers, right? So with climate change, given like the bigger abundance of hurricanes now in the Gulf region, so why do cities like Houston, Tampa, or even like, in California, like San Diego, those don't seem to have those same problems? Why is that? Like, do they have, is that a lack of data? Is there a different infrastructure that deals with this, or?
Yeah, no, they have worse problems. Their sewer systems get flooded and then everything's in the street. So Houston, and I didn't even touch on flooding, but just mass flooding in a city, you put your boots on and you're going to walk to your car. You have to know you're walking through sewage. I mean, that is exactly what happens in some of these flooded areas. There's a couple papers that showed that during Hurricane Harvey and Katrina, where they could see all these sewage signals in that water. So I think while those cities don't have combined sewer systems, and I don't think their intense rainfall, I think goes, like right to the ocean, they need a higher benchmark, like a hurricane, to really inundate those systems and have them, overflow. They probably do have separated sewer overflows. So almost any city has the ability to take their sewage if their plant is threatened and send it to the river. So it would be interesting to see, in areas, like Florida, how often do they have to do separated sewer overflows? And that's not data I've actually looked at, but I can ask one of my colleagues, how often do they have sewage overflows? Yeah, I didn't mention that mechanism. Combined sewer overflows are just, the problem is all the rainwater is going into those pipes, but all separated sanitary systems are leaky. And when it rains, treatment plants see flows go up, and in severe rain, it can get to be too much, and they'll have to essentially open a valve and let sewage be released.
Question from the Zoom, whether Legionella is present in wastewater, and if so, how common is it?
We haven't seen we see everything in wastewater, but we haven't seen Legionella as a major guy. If I was going to wildly speculate as a microbiologist, it might be a little wimpy, and not, the guys we see are like these robust microbial weeds, these heterotrophs. We do see a lot of Neisseria, so I don't know what's going on with that, which is really interesting. But we don't see a lot of, we think of Legionella more in kind of cooling water and then, being aerosolized. But now I'm going to go look at my data and look for Legionella.
Great talk. Thank you so much. So talked a little bit about the overflows and cracking pipes, which are great arguments. I was wondering about another thing that didn't show up in the talk, which is like sewer system dead ends. So for instance, when some portions of the sewer system don't get used anymore because something gets abandoned and so on, there's probably a lot of accumulation there that could probably become frequently washed off during the flooding, which would be a huge pulse. And I saw like the point pulses on your map. So I'm wondering what's your take about on that.
I don't think it's just the dead ends. I think there's a lot of kind of reservoirs of organisms, both in the combined sewer system and in the stormwater system. And unless you get a lot of turbulence and a major rainfall, you don't mobilize that. But we definitely see turbidity and a bigger visible plume go in with a higher rainfall. But what's interesting is we don't see, at least not linear, a relationship to the intensity of bacteria. A larger storm, we get way more sediment and maybe just a smaller step up in bacteria, because I think eventually these things die. They're not in kind of that dynamic, constantly fed system, but the sediment accumulates.
Okay, so I am an MPH student in policy and promotions. I am not doing anything remotely related to lab work or sequencing anything. But I'm personally interested in climate adaptation as a health issue and infrastructure issues in urban settings and how they're impacted by climate change. And so I guess I'm wondering, you're mentioning how like your techniques could possibly be used to help like prioritize where you're fixing first and where you're spending the money and those things. And I'm wondering, what are the barriers to that right now? And also, like, if you had like $1 million to do this, what would you do first?
Oh, that is such a great question. I'm really fortunate to work with Melissa Scanlon, who's the director of our Center for Water Policy. And before she was the director, I think she was a policy fellow at UW Law, and she wrote a paper using my work as a case study to study exactly that. Like, what are the barriers? Because there's such a big gap between like the research knowledge and the guys who are actually deploying monitoring equipment, you know, even those city managers, right? So she kind of proposed that you need trusted intermediaries. And that's a model that I constantly promote, because I can't talk to every municipality, right? Or the people in my lab can't do that. And we work with some long-term partners, and we see them starting to become the trusted intermediaries for other municipalities. It was so gratifying. One stormwater conference I was at, Steve Keith, who's now retired at our Milwaukee County. He just went on a 10-minute kind of, you know, explaining how all these things work. And I thought, wow, that's really cool. Like, he's kind of spreading the knowledge, but I think we need a more formal process. And I think that's where some of the NGOs really can fill that space. So we have a group called Sweetwater, and they originally, I think they've kind of, their original mission, they were looking at our TMDLs, which is total maximum daily loads. It's how we kind of regulate or enforce the Clean Water Act. But now they're finding a space working with municipalities and connecting some of these higher tools to the municipalities. So they're the go-between, all the stuff my lab can't do, you know, explaining how it can be used, setting up a study design, collecting the samples, doing the E. coli, and then they will just give us a run of samples to do our specialized part of it. And eventually, especially with COVID, these kind of molecular tests are becoming more mainstay in laboratories, especially public health laboratories. And a lot of public health laboratories can have the capability to do environmental samples. And our City of Milwaukee Health Department is kind of moving in that direction. They're doing beach testing, and ultimately I could see them, you know, doing stormwater samples for different municipalities. But I think you need that knowledge base. And I think that's going to be at some kind of agency that specifically takes on that role of being the middleman, you know, translating research.
On that note, have you worked at all with GLISA here at U of M? And they work with NOAA as well. They do a climate adaptation. They try to do more regional climate adaptation.
You know, I haven't. We have Wisconsin Initiative on Climate Change Impact. So I kind of work with that group a little bit. So I'm sure we're once removed with some of the mission
I know that
work.
they connect with other people.
Yeah, but that was a really great question, because I think we can do all this in the research lab and it just isn't going to get implemented ever, you know, unless somebody very intentionally tries to bridge that gap. Oh, what was the $1 million question? If I had $1 million?
What would you do with it?
Oh, what would I do? Well, I'd get Kentucky back in the bracket. I can tell you that. What would I do? You know, $1 million is not a lot. You know what I would do? Actually, I know exactly what I'd do. I would do student fellowships, because that is what we're lacking. I mean, I think that, and we see it in our agencies where our graduates have gone into the DNR, the Milwaukee Metropolitan Sewage District, and Sewer PAC, which is the Southeast Wisconsin Regional Planning Commission. It's like, it's so easy for us to talk with those people, because they are taking this like really, I don't want to say high end, but higher tier kind of knowledge and going into these municipalities and working. So I would say the best thing we can do is really get a savvy workforce that can, you know, start to really, you know, use these newer things in their kind of day-to-day, you know, implementation.
Thank you for your talk again. I'm curious about for I did a little bit of research with the stability in sewers for viruses, and a lot of reports said that between the time stuff gets into the sewer, by the time it reaches the treatment plant, it's less than 24 hours, and this can be even faster for rain events. But now, after seeing your talk, I'm kind of questioning that and talking about dead ends and talking about leaks. Is it that fast? Are things kind of getting caught in the sewage? Yeah, I'm curious you had a take on that. Should we be flushing our sewage? I know people talk about flushing for drinking water a lot. What would that look like for wastewater? I know it's not pressurized. So yeah, any thoughts on that?
You know, we've done a couple of different studies where we've gone from like neighborhoods to the wastewater treatment plant. And one thing we see just on the relative abundance is we see more human microbiome organisms at the beginning of the pipe. And then it shifts, the relative abundance shifts. We don't know if that's die-off or more injection of these organisms as the turbulence goes on. And we haven't really, mass balance is kind of complicated, but we have gone in and done sewer sediments, and we can find the human markers, but you know, my intuition is that they probably aren't, you know, becoming permanent residents like at least the fecal anaerobes aren't, like some of these other organisms. But what we do see is E. coli, and this relates to some of the antibiotic resistance discussions we've been having. We do see E. coli actually magnifies in those sediments. So while we see the Lactobacillus and the human Bacteroides go down, we see E. coli go up, and Klebsiella and some of these other kind of facultative anaerobes. And think how easy they are to grow in the lab. Of course, they can grow in the chemostat of pipes. So I don't know if I'm answering your question directly, but I do think the dynamic is just, you know, constant kind of resuspension of sediments, and really high turbulence will really send things up. But, you know, over time, I think it's just kind of a cycling process. And I think it is a dead end for the human organisms. Otherwise, I think we'd see evidence of, you know, way more human organisms than these other organisms if they could really take up a niche there.
Thank you.
That was great talk. And I just have a comment following up on the other question.
Yeah.
I think it's great to do student fellowships, but what I think we really need is we need some political activists. Because the problem is you can train people, but they're not going to go into public service if you get paid crap.
Absolutely.
And I think this is a real problem. And the other real problem is obviously the talk started with that we have a failed infrastructure.
Mhm.
And you know, when I look at it, it's very easy to raise money to build a new building. It's very hard to raise money just looking at Michigan. It's been a long time. People don't want to say, "Oh yes, I paid for the new plumbing in the building." On it. So I mean, this is somewhere where we need you young people to be doing what it needs to take political action, and it's going to mean raising taxes on people who can afford to pay more, and depending on NGOs, but getting government, because you need government for these actions, and government service has to pay well to have these things happen. So that's my soapbox. Anyway, thank you. That was just a terrific talk.
Yeah, thank you. Oh,
It just came in online, sorry. Is there any interest in expanding the database with African, Central, and South American information?
oh, absolutely. I think our project, the World Harbor Project, where we got 17 different cities, I think we got we were sampling harbors to look at kind of the pulse of sewage in those harbors. And we see that these markers are pretty universal, but we haven't tested them rigorously in other parts of the world that maybe don't have as much water monitoring programs. So yes, absolutely. I think there should be a good worldwide map of, you know, where these organisms are and how we can use them. That's a great question. Yeah. Thank you.
So class, please, feel free to remember that and either end, but then in the ballroom, there's coffee and snacks and I'm taking care of this opportunity. I think people and